it's a real honor to be able to introduce Dr. Ron Germain to this audience. Now, of course, it's ridiculous, the idea that I would introduce Ron to this audience. There's no need to do that. But it is always exhilarating to go back and to look at how somebody like Ron got to someplace like here. And just to recap a couple of very brief things, Ron got his bachelor's and his master's at Brown in 4 years. It did take him 6 years to get his MD and Ph.D. at Harvard. And then he went from that to be on the pathology faculty at Harvard for a couple of years before coming here. And, you know, the good news is not leaving. As you all know, Ron is really quite a visionary scientist and has always been so. From his early work on T-cell biology to his current work, really in, I think, I look at it as more lymphoid biology generally. And on the one hand, Ron has been putting together, as I'm sure he'll show you, extraordinary global pictures of what goes on. As they say, though, you know, the devil is in the details, and Ron has neither shied from the details nor failed to meet the devil and stare him down. And the work that he has done, the clarity which he has brought to it, has really changed the way that we look at these things. The other thing that is really I've respected enormously about Ron is I've had the experience of him coming in as an outside interlocutor for research groups and coming into topics with which he had no prior experience, but was able, just by dint of, you know, thinking hard and reasoning clearly, to help bring to those of us that were more benighted some guidance and some better path forward. So it's really a great privilege to introduce somebody who was the founder of the Center for Human Immunology and who has been really a major driver for immunology in the United States, in the world, and here on the campus. So thank you very much for letting me take the time to introduce Ron and to bring him to you.
Thank you, Steve. So I want to start by saying this is the Paul Lecture in memory of William E. Paul. And Bill, and that's what I'll use from now on, was a really towering figure in immunology. I'm just going to show you, he did have a younger days and very happy days. And he recalled in a front piece that he wrote called Endless Fascination how he became an immunologist, which is to read a small volume of essays by Michael Heidelberger, who was still doing research in his lab at over 100 years old, so something all of us could aspire to. He actually was doing endocrinology here at NIH from 62 to 64, but he went and became an immunologist by training with Baruch Ben-Asaref at NYU, and then moved with Baruch to NIH in around 1968. And when Baruch left in 1970, Bill became the chief of the Laboratory of Immunology, a position that he held for 45 years. I ran out of space on my slide describing Bill's contributions, and I'm not going to read this to you, and just point out that he did very early work about the difference between polysaccharide and protein antigens that in a way is the precursor for conjugate vaccines, discovered IL-4, worked out the feedback effects of cytokines that drive polarization of T cells, but did much, much, much more. He also did a lot of spectacular training, and people who passed through his lab and under his tutelage include Charlie Janeway, Ron Schwartz, Laurie Glimcher, Melissa Brown, Mark Davis, and then our own Jeff Zhu here, and our previous own Josh Milner. We actually shared the same mentor. I did my MD, PhD, and my PhD part with Baruch Ben-Asaref, who you see in the center here, with Bill and I celebrating one of Baruch's birthdays. This is obviously a staged photograph. We did not typically always wear our white coats and ties in the lab, although Bill did fairly often. But we did read sequencing gels together in the days when that's how you did sequencing. And why is this lecture titled Looking at Fundamental Immunology? And the answer is because Bill, among the many things that he did beyond the laboratory, so for example, being the primary editor of the Annual Review of Immunology for 3 decades and making it the most cited publication in biomedical science, not just immunology, is that he published this multiple editions of the book Fundamental Immunology. And that's where I really want to start my talk today, which is thinking about the fundamentals of immunology. And we think about the immune system as a protective system that defends us against the dangerous external world. And more recently, we think of it a lot in having an influence in protecting us against a dangerous internal world. And the question is, how does it do this? These are very diverse, unpredictable disease entities that arrive or arise at unanticipated times in different sites of the body. How do you survey everything and know that you and are able to recognize what's going on? And to meet this what, when, and where challenge, the immune system uses soluble effectors, and I'm not going to talk much about them today, but antibodies, complement components, pentraxins, and so on. Mobile elements, and those I will talk about, that can move directly into the relevant affected target site, and organized tissues, and I'll talk a lot about this today, that are necessary in order, especially for the adaptive immune system, to do its job, given that it originates its response from very rare cells in a very large volume of the body. And so we have cells that are formed in the bone marrow, and these are myeloid and lymphoid cells. The more mature myeloid cells are ready to go when they exit, and they can find inflamed endothelium. They can adhere, roll, and transmigrate and exit into the tissue to mediate effector function. Some precursor cells that come out of the marrow can enter the thymus and go through a whole developmental and selective process to give rise to T cells that wind up back in secondary lymphoid tissues. For example, this lymph node. Within the lymph node, there's a segregation between these T cells and another form of lymphocyte, the B lymphocyte here in these follicles. They eventually have to get together. And I'll point out that immunologists have never figured out why they need to be separated before they get together. Something to think about. But within the T zone, the T cells are searching for their cognate ligand, and that ligand is typically in the form of a peptide major histocompatibility complex ligand on the surface of a dendritic cell. If there's a productive interaction where that ligand matches the specificity of a clonal receptor on the T cells, those cells get activated, they can proliferate, differentiate, and then like the innate cells, they find inflamed endothelium and enter into the tissue to mediate effector function. And if we think about what I just described, after especially the cells leave the bone marrow, everything that they're doing when they're not in the blood per se is in a tissue. They're either becoming activated in the tissue or they're mediating effector function in the tissue. And so we cannot understand how the immune system does its job if we just look at isolated cells and molecules. We will never understand exactly how this system works. And in order to look at what's going on in a tissue, we need to image, and we want both dynamic and high content information. And so I'm going to start with where we and others in the field started about 20 years ago with the dynamic imaging. This is multiphoton intravital microscopy. It reveals very nicely temporal aspects of biology, but it's typically limited to 5 or fewer parameters, and that's a major limitation, as we'll get into later. It's possible to do signaling measurements, but it's typically pretty much calcium or the analog of NFAT nuclear translocation. You can look for a few seconds to hours. There are window preparations where you can look over longer time periods, but then you're not following the same cell over all that time. You're looking at iterative populations of cells over the timing, and it's not really applicable to human studies. You can do explants, but there's no innervation, there's no lymph flow, there's no vascular involvement, there are no cells coming in and out in the normal way, and so this is a very limited application. So the way you do this is surgically expose the tissue of interest. You only get a free lunch in the ear skin where you don't have to do surgery. And of course, surgery is disruptive and pro-inflammatory, so you should ask when you read papers using this method, how did people control for the background inflammation that they cause by doing the surgery? But assuming they did that well and properly, you take a laser and scan it in the XY dimension, get a plane of imaging information, focus up or down, get a stack of information, and then collect all of that over time. So 4-dimensional data sets that you play back this way, 200 to 300 times real speed, and compressed in 2 dimensions. But we do have the 3-dimensional information. And here are T lymphocytes moving in the T cell zone of a lymph node, and it looks like they're free to move around like a goldfish in a little bowl that you bought at the state fair, but that's not true. They're in this environment. The black is not empty space. The black is just what we don't make visual by putting fluorescence into our images. And so you should never believe what an imager tells you based on what he or she does not see, and you should always worry about what's in the black, because it is influencing the behavior you're visualizing, and it's not to be ignored. So I think of the immune system in sort of 3 major components: the barrier, the skin and mucous membranes. If you breach them, you get a rapid innate immune response, and then more slowly develop an adaptive response. So I'll talk about 2-photon imaging in the context of innate immunity. And this is work done by a spectacular colleague who now has his own lab in Erlangen, Stefan Uderhardt. And what Stefan wanted to do was to observe myeloid cell behavior, and to do that he chose to do it in a new preparation looking in the peritoneal wall. And the reason for that was we know there is this monocyte-derived population of peritoneal macrophages, but there is also these embryologically derived fixed tissue macrophages in the serosal lining of the peritoneal cavity, and then it's easy to recruit neutrophils with a challenge. So we could look at all these different myeloid populations at one time, and when he did that, he took advantage of a model that Tim Lammerman had developed in the lab earlier, where we did sterile injury with laser damage in the dermis, and what he did is do the same type of damage in the peritoneal wall. And when he did that, what he saw was the same type of neutrophil swarming that you see here. You see that the corpses that were in white here are destroyed and phagocytized or pinocytized by the neutrophils, but this is the problem. Here's the collagen that's part of the tissue matrix, and you see that the neutrophil swarming damages this matrix. And that raises an interesting question. Cells die in tissues all the time. If the neutrophils respond to cell death by swarming in the way that I just showed you, and then they cause this collateral damage, how would you ever maintain tissue physiology? Because you'd have this constant inflammatory process that was disrupting the tissue structure. And that did not make any sense, so we needed to understand what was preventing this, or we presume was preventing this. And so what Stefan figured out is we had always killed masses of cells with our laser. And he changed this and killed a single fibroblast in the peritoneal wall. And that's what you see in orange in the middle of this image here. And then he observed what happens with genetically labeled myeloid cells, and here we're looking at those embryologically derived macrophages, and what he saw is they rapidly sense that cell death, and then they don't move the cell bodies, but they send out processes that encompass the corpse, something we call cloaking. And that turned out to be the key feature. So normally, if we damage multiple cells, we can see this kind of tracking of the neutrophils. They sense it, and then they all turn and run in a very directional way to create the swarms that I showed you in a moment ago. But if we have the single cell death, neutrophils do show up in the preparation, but they don't show that highly directional and rapid migration. And the reason they don't is they can't see the corpse. And what Tim Lammerman had previously shown is that the swarming is initiated by a lead neutrophil sensing and actually touching the dead material and beginning to produce the leukotriene LTB4, which then induces other living neutrophils to make LTB4, creating an enormous gradient for these cells to follow to generate the swarming response. And it turns out that this protective mechanism is critical. If we chronically deplete these fixed tissue macrophages and look at a tissue that's undergoing chronic stress, the diaphragm, you're breathing all the time and stressing the muscle fibers, now what we see is we get these necrotic muscle fibers when those macrophages are not there to cloak, and these are associated with very intense neutrophilic infiltration. And so what Stefan's work showed is that the resident macrophages prevent innate immune damage in response to intermittent cell death or damage, and it permits healing by local replacement. The neighboring fibroblast divides, fills in the gap. There's no resolution, a post-inflammation type resolution, and there's no scarring. So it's very benign for the tissue. If we do very careful work over time and in aging animals, we see that these rather protective macrophages begin to acquire more inflammatory properties. And so we think that this process may be subverted and contribute to what's called inflammaging. And it may also be that chronic stimulation of things like myeloid in the brain cause microglial cells to move from this more protective state to a more inflammatory state that contributes to diseases like Alzheimer's. And so this kind of live imaging has been very valuable, letting us see things that you could not possibly see with extracted cells in vitro conditions or by single cell analysis. And we've used it to look at dynamics in lymph nodes, for Mike Bajanov to discover that T cells move on fibroblastic reticular cell roads to find their dendritic cells within the T cell zone, to look at dynamics in germinal centers that are making antibodies, to look at dendritic cells reaching into the gut lumen to sample the microbiota, as work that Jackson Egan did looking at function in the kidney, and also work that he's done with mycobacterial granulomas at both the myeloid cell and the lymphocyte level, to look in the lung as Parazad has done at inflammation in response to influenza or tuberculosis infections, to look at these rapid swarming responses of neutrophils in the ear, but also at T cells responding to delayed type hypersensitivity inducers, and as Masaru Ishii did in the lab, to look in the bone marrow at osteoclastogenesis. So it's a very diversely useful approach. It's been applied in many different tissues. I want to give you a sample of the way we've used it over the years, but there are many other laboratories who've used this in their own ways. And the problem is that here we see in blue a bilobar mouse lymph node, and we only image this small volume. And so we had a lot in the black, and I told you need to worry about what's in the black. And so I encouraged the people in the lab to do some histology, to section the lymph nodes and see what we weren't seeing in our 2-photon imaging. And the problem was, when we went to do this, we realized that the methods for doing staining were 2 or 3 color. Typically you did a red and a green stain, maybe you looked for yellow to see if they were coincident. But most of the people in an immunology lab by this time were doing flow cytometry experiments between 9 and 15 parameters, because it took 2 to 4 parameters to identify any given subset of cells. So 2 colors obviously didn't cut it. And so we decided we needed to move to multiplex static imaging. And here we're not going to get these real-time dynamics, but now the parameter space, instead of being limited to 5, is easily 30 to 40, and we can push it above 80. We can look at signaling and post-translational modifications. We can look at proteins and RNA. And obviously, we can look at ethically collected human material. And so we started with what we called histocytometry a bit more than a decade ago, and this is work that Mike Gerner pioneered in the lab, and Mike has his own lab at the University of Washington now for a number of years. And the way this worked is we've stained in with multiple fluorescinated or fluorochrome conjugated antibodies, and do confocal imaging. We might deconvolute to improve the quality of the images a little bit. We'd compensate for any channel spillover like you would do in flow, use software tools to segment objects with their associated fluorescence. But those data are equivalent to list mode flow data, cellular objects with fluorescence. So we could then put that information into flow cytometry software and get quantitative data where we're gating on multiplex expression of marker sets. But since we kept the X, Y, Z coordinates of anything we might gate, we could then identify precisely where those cells resided in the original tissue. And this method works not only with so-called marker antibodies, meaning mostly self-surface proteins, but also antiphosphoprotein antibodies and some anti-cytokine antibodies. And it can resolve the difference between localization of transcription factors in the cytoplasm versus nucleus. So then it means we can look at cell state, at cell function, and phenotype all in the context of spatial positioning in the tissue. And this is information you cannot get even from modern spatial transcriptomic methods that only look at the RNA. The signaling components, for example, are invisible under those conditions. And so I'm just going to summarize in some words here what combining some of the earlier two-photon imaging with histocytometry has taught us. And there are two very important lessons. One is there's a very polarized distribution of innate cells at barrier sites that enables the system to sort of head off the invasion of the infectious agent before it gets into the systemic circulation, and that's critically important. And the other is for the adaptive immune system, these cells are not just generally co-localized in a tissue and then randomly dispersed or moving around looking for each other, but they actually are very, very precisely organized to facilitate the rare interactions you need to drive T or B cell immunity. And so I'm going to tell you one story that goes with the first of those two major conclusions, which is very recent work that was done in a lab by Anita Gola, who's now working with Elaine Fuchs at Rockefeller. And this has to do with the liver. So the liver is a bit unusual in that it has two circulations. It has the portal circulation bringing essentially nutrients from the gut into the liver. And then it's got the normal circulation. There's a hepatic artery. And then both of these inputs then flow out in the hepatic vein. And if we scale in, we can see that a liver lobule is set up where there's what's called a portal triad that has the portal vein, the artery, and a bile duct. And the flow is toward the central vein through sinusoids that are lined by a number of lymphoid compartments, mostly NK and NKT cells, and these myeloid cells, these Kupffer cells that are members of the macrophage lineage. And then you have the endothelial cells, forming the edges of the sinusoids. And it's well known that the liver is metabolically zonated. There are different things that happen toward the periportal region versus happens toward the central vein. But the literature at the point that Anita started these studies said that's not true of the immune system. Basically, the T cells wander in a random way, surveying what's going on in the sinusoids, and the display of those other cells that I showed you, especially the Kupffer cells, they're just distributed along the sinusoidal walls. So here's, going back to the two-photon imaging, the behavior of NKT cells in the liver, which is originally described as random behavior. But if you look at these tracks, you see that they're concentrated here near the E-cadherin-rich staining areas, and these constitute the periportal region. So there's not random motion. The cells move out away from the periportal region, but then run back, almost like a yo-yo going out and in, and spend most of their time in the periportal region. And if we then look at the Kupffer cells, when I superimpose the E-cadherin staining near the periportal region, and I'll just go back again and do that, what you see is almost all these red F480 stained Kupffer cells are within that staining for the E-cadherin, and they're not in the central region, which is where the central vein is. There's a very strict polarity to this distribution. And I'll just skip over some of this more quantitative data. Well, is that, in essence, inborn, ontologic in the same way that the metabolic zonation is? And the answer is no. If we look at a newborn, we don't see that zonation at all for the Kupffer cells. And then as the animal ages, we see a major transition at around day 20. Well, day 20 turns out to be when you wean the mice, and they markedly change their microbiota. And so we asked, does it matter? And yes, if you look in a germ-free mouse, now you don't see the polarization. If you look in germ-free animals that have been co-housed, their Kupffer cells polarize in their distribution. And if you take an SPF mouse with polarized Kupffer cells and treat it with antibiotics, they lose the polarization. So all of this told us that it's the microbiota and signals from the microbiota that matter. This is not something that's built into the system ontologically. And in coordination with that, in agreement with that, if we get rid of a major adapter protein that's critical for signaling through toll-like receptors, as well as through for IL-1 and IL-18 signaling, MyD88, then we lose this asymmetry, and we could show that knocking out things like TLR4 had partial effects. So we're quite confident that this is what's called PAMP signaling. That is a response to things like endotoxin that are released from the gut microflora and highly enriched in the portal circulation, being carried to the liver and entering in that portal triad region. That's the highest concentration, and then flowing out. And what we then showed is that what those signals do is they don't talk directly to the Kupffer cells, they talk to the lymphatic endothelial cells, and they instruct those cells not for how much chemokine they make that guide the Kupffer cells, but how they reconstruct the extracellular matrix of aminoglycans to which the chemokines bind. And so the gradient control is based on extracellular matrix remodeling that's responsive to these signals. Well, why is the system polarized this way? And so many years ago, Jackson Egan did this experiment by 2 photon. In green are the Kupffer cells. In blue, there's a tracer that shows the blood and the sinusoids. In white are going to be bacteria that are put into the vascular tree. And what you see is when those bacteria hit the liver, they stick directly to the Kupffer cells. And we created a mathematical model that said if we make all the Kupffer cells equally sticky to bacteria, because that's something we can't easily control if they do vary in their stickiness across the sinusoidal distribution, what happens if we have them polarized versus non-polarized? And the answer is when they're highly polarized, the bacteria are captured very close to the periportal region and very few reach the central vein, meaning going into the general circulation. But if we make them uniformly distributed, now lots of the bacteria make it to the central vein and go into the circulation. Well, this was a model, but Anita did a heroic experiment of injecting bacteria directly into the portal vein. And when you do that, you get exactly that result. In animals with the polarized Kupffer cells, most of them are trapped very close to the periportal region. Very few reach the spleen or the blood, here in black. But if you use a germ-free animal with a non-polarized Kupffer cell distribution, now you see lots of bacteria making it into the circulation, being captured in the spleen or present in the blood. And so that gives us a model for what's going on here, where this polarization is meant to enable the system to most efficiently capture anything that's escaping the mucus and epithelial barriers in the gut, getting into the portal circulation and potentially going systemic. And you need that polarization of the immune system to do that efficiently. And although it's not absolute, when you think about these bacteria chronically coming out of the gut, if you weren't doing this all the time, you'd be at great risk for systemic septic events. I think I've lost my control here. There we go. And so I'll just say that that fractal biology, as I call it, also applies to lymph node structure and function. We don't have lymphocytes everywhere. I mean, they're in the blood, but they're not in all the tissues. They concentrate in a lymph node or a spleen. Within, say, a lymph node, they separate into those T and B areas. In the T area, you have this fibroblastic reticular network. The T cells walk on that network, and the reason they do that is it reduces their search volume looking for dendritic cells that are on exactly that same network. Within the context of this network concentration, if you get an engagement of one T cell with a dendritic cell, it makes chemokines that then bias the walk on the fiber network to direct those cells to the interesting dendritic cell. And then Antonio Perez in the lab showed that there is a polarization match between dendritic cell subsets and T cell subsets. They're not randomly dispersed within the T cell zone. And that polarization is critical to the right timing for activating the different subsets of lymphocytes. And if you destroy that polarity with no other change in the lymph node, now CD4 cells don't cooperate properly with CD8 cells to make good memory in response to vaccines, for example. So it's this organizational level is absolutely critical to adaptive immunity, and it's both structural organization and chemical organization, just as I showed you in the liver. So we've now applied this histocytometry to cancer biology, and we is actually Vivian Maltese in the lab. And she's worked together with Caitlin Byrne, initially at Penn and now in her own lab at Oregon, in a model in which it's a genetically engineered model of pancreatic ductal adenocarcinoma. And what Kate did was to clone individual cells from those primary tumors. And it turned out that they distributed into two groups, what we would typically these days call cold or hot tumors in terms of the T cell infiltration and the bias toward suppressive macrophages. And so what Kate and Vivian did was to study animals that are bearing a hot tumor with a treatment that combines checkpoint therapy with an agonist anti-CD40 antibody, a combination that's in current clinical trials and is highly effective in this animal model. And here they were looking, and Vivian in particular was looking very shortly after the combination that involves the anti-PD-1, anti-CTLA-4, and the agonist anti-40. So this is only a 2-day window. And what's striking when this is done, looking here at a dot version rather than the fine-grained primary analysis of the images for T cells, is that there is a major loss of FOXP3 positive T cells, so what are called regulatory T cells. And the few that remain, and you see how much this reduction is here, are in the periphery. And that's going to be important when we draw some conclusions a bit later on. And this just quantifies that change in distribution. Now, it seemed like that was associated uniquely with the anti-CD40, and this experiment shows that that's true. If you use a CD40 knockout animal, this treatment does not deplete the the regulatory T cells. But that was very surprising because CD40 is not expressed by regulatory T cells. So why would this antibody give you this phenotype? Well, CD40 is expressed by dendritic cells, and if you get rid of a particular subset of dendritic cells in a transcription factor knockout, a BAT3 knockout, now this treatment does not cause the loss of the Tregs. So the question is what's happening to the Tregs, and why are dendritic cells involved in losing FOXP3 positive T cells? Vivian did a number of experiments looking to see if there was any evidence the cells were dying in situ or being phagocytized, and the answer is that are basically negative data. But it led us to wonder whether these cells were losing FOXP3 expression rather than being eliminated as T cells entirely. And to look at that, we turned to lineage tracking, and you do that in an animal in which you can give tamoxifen to do inducible turn-on of the rosa tomato locus so that you get the red fluorescent protein expressed in cells that are expressing CRE, and the CRE here is under the control of FOXP3, so only the authentic Tregs are going to show us this tracking when we do that. And then you go through the regular procedure, and the surprising result, the very striking result is, as you see here, we get the elimination of most of the Tregs and the residual ones being on the periphery of the tumor, as we saw before. But now we get a very large population of so-called X-Tregs, cells that have the lineage marker but no longer have FOXP3. These are CD4 positive, CD3 positive T cells, and they also populate the periphery. And this just quantifies those changes. And it was verified also by flow cytometry. So how does that work? Well, the dendritic cells that rely on BAT3 are a subpopulation that Keitano Reis-Souza and Alan Scher showed years ago are a major IL-12 producer, and anti-CD40 induces the P70 form of IL-12. And so Kate and Vivian looked at whether IL-12 and the cytokine that's promoted by IL-12, interferon gamma, mattered. And as shown here, if you treat with either anti-12 or anti-interferon gamma, you no longer lose the majority of the conventional FOXP3 Tregs, and you don't make nearly as many of these X-Tregs. Well, okay, there's the conversion and loss of FOXP3 is dependent on 12 and interferon gamma, but this goes back to Bill Paul. Those are cytokines that induce cells to become Th1s. So is that happening to these cells as they lose the FOXP3? And here Vivian turned to another technology for looking at targets in the tissue. Here it's RNA fish. And I'll just show you the blown up part, which shows you that these X Tregs are actively making interferon gamma message, and flow cytometry confirms they're making interferon gamma protein. And this is without any restimulation. And so what's happening here is that the anti-CD40 action, probably together with some conventional CD4 cells, gives you a combination of IL-12 and interferon gamma production, which causes the loss of FOXP3 expression in the conventional Tregs. I didn't show you this, but they also gain in T-bed expression. And they become type 1 effectors that actually make interferon gamma, which is a critical cytokine for anti-tumor responses. It's considered a canonical outcome of doing checkpoint therapy. But in this model, the checkpoint therapy alone doesn't do that, even in these hot tumors. You need the anti-CD40 as well to get this at a high level. In addition, the movement of any residual Tregs to the periphery removes them from proximity to the responding conventional cells, which then limits their ability to suppress the response. And so overall, you get a very potent anti-tumor effect because you're at multiple levels removing the suppression within the system. But not only that, you're actually adding to the effector function that's attacking the tumor. Now, the histocytometry has been obviously very valuable in many different ways, but it's not adequate. There are many more immune cells, subsets or types than I were able to look at in the number of colors that I showed you, and number of parameters. There are all these epithelial and stromal cells you want to look at in the tissue at the same time. There are acellular structural components, and if we're interested in tumor biology, there are other things going on with the tumor cells. And so Andrea Radke did a spectacular job in the lab of setting up a new iterative staining method, which is called IBEX. You stain in somewhere between 4 and these days maybe even back up to about 8 or 9 colors. You image, you bleach, and then you iterate this process. And then she worked with Ziv Yanov and Brad Lohnkamp to create ways of registering all these iterative imaging cycles, because there are small differences as you go through this process. So we get pixel level resolution here, and then we can go into other software I'll talk about for doing analysis. And so here's an example of a human lymph node stained with 82 different antibodies, of which 60 some odd actually react with molecules in that environment. And then that really raises a question. You could look at combinations of 2 or 3 markers and sort of perceive different patterns, but how would you look at 62 different parameters at any given time and getting some understanding of what's happening in this tissue environment? And so this is a challenge taken on by Nishant Thakur and then with help from Spencer Grant in the lab. And what they've developed is a software tool called RAPID. It's based on machine learning. And it basically does a complex analysis to sort of cluster the different fluorescent pixels you get in these images into things that more realistically represent what you would see if you were doing multiplex flow cytometry. Now, the pixel approach is important because all segmentation algorithms fail at a certain level with very irregular cells, and they're not very good at extracellular acellular matrix components. The pixels don't care about those, and they're much more useful. But built into this software is also the ability, when it's useful, to do segmentation. And we've compared the segmentation algorithm here to other models, and it's as good or better under all the conditions that we've tested. So as an example here, with Andrea's human lymph node data, here's the imaging picture. It gets translated into the pseudo color in RAPID. And what you see here is a germinal center. And the pseudo colors are not segmented cells, but they are representative of a cluster of pixels that the software has decided are unique and different from what is adjacent to them. And that winds up looking like individual cells. And if we go and ask what it's showing us, it's using a combination of 4 different markers to look at naive B cells that live outside the germinal center, 6 different markers to look at germinal center B cells, and you can even see in the pixel image the shape difference between the light and the dark zone B cells, to look at which of those B cells are proliferating, to look at TFHs, and so on. We don't all want to write Python code, and so Nishant and Spencer have turned this into a graphic user interface, so everything is point and click. I'm not going to have time to show you all of how it works, but just to say that you can load in your image and do some manipulations of it. You can then do either object or pixel-based segmentation and clustering analysis, so you wind up with information looking like this on your screen. And you also get these tables that quantify what the cluster and pixel calls are in the software. And so here, for example, the only strong signal with is CD31. That's representing endothelial cells. Here we have a combination of high CD3 and CD8. Those are CD8 T cells, but if you look at 62 parameters, maybe those CD8 T cells are CD44 high, so they're not naive, for example, versus the ones that are CD62L positive, and so on. And then you can do a limited amount of spatial analysis. But as I told you for histocytometry, this is the input for really beginning to look at what's happening in space. And I say that for a reason. And there are all these different possible patterns across tissue scales. The zonation I already talked about, the broadcasting of signals based on cytokines, or physical cellular entanglements of two or more cell types. And so Ed Schramm has done a spectacular job of approaching this as a mathematical problem, creating another software tool called SPACE that enables you to look ab initio at what's happening in the tissue with all these different parameters. And so this first step is to go into RAPID and do this kind of clustering that I've already told you about. And then you do this iterative census of local neighborhoods. It is not a neighborhood algorithm of the type that you may have read about in the literature. It just uses this to gather information across the entire tissue space. And then he goes into a mathematical approach called generalized mutual information that determines whether there is a statistically relevant asymmetry in the distribution of various components in the tissue. And from that, you can learn relationships. And I'm not going to walk you through all this. This is still, being finalized, but I'll give you an example of how powerful it is. So here we're looking at a 42-plex mouse lymph node that had been not analyzed previously for this particular image, but had been studied previously by Antonio Pires in the lab. And what he was able to show, as I mentioned previously, is that the CD4 and CD8 T cells are biased in their distribution in the T cell zone, and that bias matches the different distributions of CDC1 and CDC2s that are preferential presenting cells to those two different T cell populations. And what SPACE reveals over here on the right-hand side is that, of course, B cells are separated from the two types of T cells. You see that in this reduced dimensionality plot. But further, that there is a difference between where the CD4 T cells and the CD8 T cells are. And if we do that in the graphic form, you see that the CD4 cells are more peripheral than the CD8s, exactly what Antonio found after years of careful study by histocytometry, but something that comes out automatically from applying SPACE. And the current local neighborhood algorithms that are used with codecs and other multiplex imaging tools will never reveal these types of relationships because they're all based on nearest neighbor analysis of proband cell and the cells that are touching it, whereas this can look across all these scales. Now, that's very nice, but I told you earlier on we had a problem looking at a small volume by 2P live imaging and looking at any given section through a tissue also under samples. And so Weijie Li and Mike Gerner got together and said, well, let's look at a volume rather than a single section. And they developed a new way of clearing the tissue, and you see the way that they've made the tissue transparent. And I want to point out that before we did this, there were many methods already published for creating cleared tissue. It included Clarity, Disco, BABB. The reason we, quote, reinvented or invented a new approach was none of those enable you to do high sensitivity direct multiplex staining. You can track fluorescent proteins very well if they're very bright. You can do a limited amount of staining with an indirect reagent for amplification. But if you want to do multiplex, you needed a different method. And that's what they developed. And so here's an example of a lymph node, completely intact, no sectioning, stained in 5 colors. And you see the beautiful arborization of the thick vessels, the HEVs. You see the full volume of those peripheral B cell follicles, but of course we can look in any given section and treat it like histocytometry. And so we get all of this information in 3D in a way that an individual section would fail to give us things like the ramifications of those vessels. And so Harry, who now has his own lab at Raygon at MIT, did a spectacular job combining histocytometry and this 3D multiplex imaging to study regulatory T cells in tissues. Jiddu Liu had shown previously, using antiphosphostat staining, that contrary to some expectations, regulatory T cells didn't keep autoreactive T cells from becoming activated. That's not how they really prevented autoimmunity. You could see that, that there was the production of IL-2, so full activation to cytokine production, from what we could show based on germ-free mouse analyses were self-reactive T cells, but they were surrounded by these pSTAT5 clusters of what Jiddu identified as regulatory cells. So we knew that these little clusters seemed to have a relevance to controlling responses, but we didn't know how they did it. And that's the question Harry took on. So the first thing he did is to analyze things. It turns out looking for IL-2 is rather difficult. He found that PD-1 expression in the steady state reflects these autoreactive cells, and we see the same number of those PD-1 positive CD4s in germ-free mice. And these are all pSTAT5 negative, as Jiddu had shown before. He worked out with Kim Young Park spatial statistical methods that allowed him to look at that proband PD-1 positive CD4 T cell relative to the surrounding populations of regulatory cells. And when he did that, what he found is that there is a clear clustering of regulatory cells around, uniquely, the PD-1 positive activated autoreactive cells, as opposed to all the other CD4 cells, the PD-1 negative cells, in the population. And I want to point out here something that, because I just came back from a meeting where this wasn't clear to everybody doing spatial analyses, you need to go and do random sampling and essentially Monte Carlo randomization of all the cells within the volume to know whether something that looks like an interesting association is really just random or is real. And because he did multiplex imaging, he could actually begin to characterize the state of those Tregs. Again, something that would be difficult to do if you had just taken everything apart and done single cell, and is beyond the resolution of the current spatial transcriptomic methods. And what he found was that the cells that were clustering near these autoreactive cells were pSTAT5 positive, Ki67 positive, CD25 high, CTLA4 high, and FOXP3 high, meaning they're highly suppressive effector Tregs. And experiments are not showing you, he showed that this is IL-2 dependent, consistent with what I showed you from Jiddu, that these cells are surrounding an IL-2 producing cell. So it told us that Tregs were operating in a negative feedback mode. They were responding to the response of the autoreactive cells. They were sensing that those cells had become activated, they were making IL-2, and the Tregs needed to do something, and what they needed to do is to become suppressive. And that's what was happening here. Now, I said they were Ki67 positive, and again, that was highly enriched in the Tregs very close to the responding cells. And so the question was whether the density of these Tregs was a chemokine attraction, or whether it could have been just proliferation locally in response to the IL-2 that the autoreactive cell was making. And so to look at that, we use what are called confetti mice that have a way of giving you a kind of clonal picture of cells based on color expression. And you can see these small clusters of like-colored cells within these Treg clusters. And if you look at that statistically, it's clear that this is non-random. So what was happening is that there was proliferation going on, And so there's a feed-forward amplification. The system is operating in a negative feedback mode and responding to the IL-2, and when it does that, it feeds forward in an interesting way by proliferating these highly activated regulatory cells. That still didn't answer the question of how this controls autoimmunity, and for that we needed the clearing and the C3D approach. And the reason is we know that if you disturb the ratios or numbers of cells within an animal or a host, you imbalance the Treg and autoreactive components. And so we needed to look at physiological numbers of cells to really address this question. Well, if we try to do that with a thin section, we might see one, if we're lucky, cell in a section. It would be very hard to get statistics. But if we looked at an entire cleared lymph node, then even 10 or 15 cells began to be statistically meaningful. And so that's what Harry did. He worked out a way of transferring incredibly small numbers of a transgenic cell we could track so that it was at physiological frequencies of a few cells per entire lymph node. And when he did that, he could see in this lymph node, in the red cloud are all the endogenous regulatory T cells. These few spots are the autoreactive transgenic cells or control cells. And he could quantify what happened over time. And there was a very big surprise. For the first 3 days, those autoreactive cells proliferated even in the presence of a full cohort of regulatory cells where we knew these animals would not get sick. But then, after a few more days, they essentially all died off. And so the way the regulatory cells work is not to prevent the activation of self-reactive cells, not even to prevent them from proliferating, but to cause them to die when they do that. And so the way this all works out is that we believe that positive selection in the thymus gives us the conventional cells at a somewhat lower self-ligand avidity than the regulatory cells. When they meet up with their self-antigen on a dendritic cell, you get a little bit of CD25 expression on the conventional cells and a little bit of IL-2. The Tregs, when they see their ligand, put up more CD25, and they have a preferential capacity to capture that IL-2, in essence, to steal it away and not let autocrine recognition occur among the conventional cells. At the same time, those Tregs that are capturing the IL-2 proliferate to create this very suppressive zone to make sure that the conventional cells don't, quote, escape and see their IL-2. Why does that matter? Because IL-2, well, is usually thought about as driving T cell proliferation, but it doesn't do that. It enables proliferating T cells to survive. And so what happens here is that the conventional cells go into proliferation in part through MYC upregulation due to TCR signaling. They make IL-2, which they would normally use to survive that proliferative burst, but the Tregs are stealing all that IL-2, and so the proliferating conventional cells don't see it, and they die, pruning them from the repertoire in a continuous basis and preventing autoimmunity. And so with Kim-Young Park and John Sang, we've created a mathematical model, and that model shows that small changes of less than twofold will dysregulate the system I just described. So for example, 50% levels of CTLA-4, a very abundant molecule on regulatory cells that steals co-stimulatory molecules 80 and 86, was predicted to allow escape from this regulation and for the conventional cells to see their own IL-2, and that's what we saw experimentally, and that matches the phenotype of heterozygous humans where CTLA-4 heterozygosity, or the equivalent in a mutation in a protein controlling CTLA-4 expression, gives you autoimmunity. And so this begins to give us a clue about how GWAS hits, things that are EQTLs of in the order of twofold differences, can begin to favor an autoimmune state. And so very recently we've been collaborating with Alex Marson, who has identified enhancers that operate uniquely to control CD25 expression, the IL-2 receptor alpha chain expression, in regulatory cells or in conventional cells. And when you do that, what he sees is that if you modify the enhancer that works in the regulatory cells, they have a lower steady state level of CD25, but that goes up to the normal level within 6 hours of triggering them through their TCR. It's a very subtle difference. And likewise, for the conventional cells, they take a little bit longer to upregulate their CD25, but they reach the full level. Well, in the case of the enhancer that works in the conventional cells, now in the NOD model of autoimmune diabetes, if you disadvantage these conventional cells for their CD25 expression relative to the normal state, none of the animals become diabetic, and that's true even if you treat them with anti-PD-1 that normally gives you this incredibly rapid and fully penetrant disease state. And conversely, if you encumber the Tregs slightly, now disease happens much more rapidly and in a more penetrant manner. So these very small changes that I showed you from the staining translate into major differences in the biology, and it shows you that the Treg system is at one level robust, but it's also very fragile, which is exactly what the mathematical modeling showed us, with a few parameters that have to stay in a very narrow range for the system to work. And so this begins to get at how genetic variation actually causes human disease. So I want to end by saying that, we always want more, and more is to go from IBEX multiplex and C3D volume and put them together. And it takes a team, it takes a village to do this. So Hiroshi, Andrea, Colin, JL, and Armando are working on this, and we're pretty close to having this resolved. The way we do this is a combination of IBEX with iterative cycles that involve the clearing with the C3D method. And here's a lung image this way in a couple of 100 microns by Hiroshi in 20 parameters. And you really begin to see that this gives you all sorts of information you don't get when you're doing low-plex or volume. And I'll let this play a little bit more and just point out that this is especially valuable not only for vessels, as I showed you in the lymph node image before, but, for example, nerves that are very hard to see in thin sections. Here's another 20-plex in the gut, so these will change with 4 or 5 colors in each cycle as this rotates, just to show you the kinds of information you can get from doing this. And so I'll end by saying that we really think that the way to understand immunity, as I started out in the very beginning saying, is high-dimensional analysis of tissue architecture, content, and cell state. We can do this by 2P imaging, by histocytometry, but we can improve that with IBEX. We can do histocytometry in 3D, but then we can combine all of that to C3D IBEX. We have software tools, Rapid and Space, for taking advantage of the information we get and quantifying it and putting it in context. And I haven't had a chance to show you, but we're beginning to combine this with RNA probes so that we can take advantage of that approach and use the staining part for those things that are uniquely approachable that way, like the phosphoproteins, nuclear translocation, and so forth. And let me end by saying that I don't have the traditional slide with 1,000 names on it. I try to tell you who's been my colleague and collaborator throughout in all the work we've done. But I want to thank everybody who's been in the lab, because there have been all different levels of contributions besides the people I've named. We obviously have had a lot of collaborations, and I pointed those out along the way. And we've really been generously supported by the intramural program of NIAID. We have also had support over the last few years for what we call CATI, the Center for Advanced Tissue Imaging from the NCI, some bench-to-bedside support, and some Jan Zuckerberg support. Thanks very much.
Oh, Ron, I love the model for the Tregs. I was wondering if, presumably that model works best in the lymph node, and what about in the peripheral tissues? How do you think the Tregs would work in the same way in the peripheral tissues, or it would be some other mechanism?
So I always leave something for the fellows that leave the lab to do. And actually, Harry is making a major effort looking in the tissues now using these same approaches. So I don't know what he's found. He's just started the lab. He's looking at it. There is reason to think in some cases that it still could be IL-2, because there is the checkpoint therapy, for example, works by expanding cells from these stem-like memory cells. And maybe that's dependent on 15 and not just 2, and then the rules would be somewhat different, so I don't want to push for this. But given that there is proliferation under those conditions, they again could operate in part by stealing. They obviously, I didn't go into all of it. The model includes the CTLA-4 effect, because it's not just stealing the IL-2, but it's limiting how much IL-2 is made, because you're interfering with co-stimulation. Well, that's going to apply in the periphery as well, obviously, and we know you need that co-stimulation for many of the cytokines that would be produced. So I think parts of the model will apply, but I don't think IL-2 per se will be as critical in the periphery.
Thanks.
Hi, very impressive imaging. I have 2 questions. The first one is regarding the architecture of the liver. How complex does the microbiome need to be for the Kupffer cells to be polarized? And the second question would be regarding the X-Tregs. Do they remain suppressive?
For the first question, we can get at least a partial effect by using LPS when they're not polarized, and we can lose a lot of the polarization removing only TLR4. So I don't think the microbiome has to be enormously complex, but it's clearly there are multiple different inputs because we get a difference between MyD88 knockouts. We didn't do TRIF and just the TLR4, but TLR2 knockout also had an effect. So the complexity matters, but it's not essential. As far as the XT-regs are concerned, we haven't traced this out over long periods of time. So we know in that acute period they convert and they spontaneously make interferon gamma in vivo. When I say spontaneously, let me correct that statement. I didn't show it to you, but we have a way of validating whether a cell is engaging in TCR signaling in situ in the tissues now. And the population of T cells with the highest fraction of cells showing that signaling state are the XT-regs. But whether they peter out, die, beyond that, I can't tell you yet.
Thank you.
Let me squeeze in one from home here. Our friend Howard Young asks, how do you deal with the data storage? It must be enormous.
Well, our institute is very happy to support large amounts of storage, so we just keep dumping it into the H drive. That and a lot of hard drives hanging around.
Hey, Ron, that was beautiful. So I know in Harry's work you were looking at steady state to look at autoreactivity, but have you started to do that in the context of an infection where it just sort of related to the question of is IL-2 rescuing lower affinity cells and how do Tregs fit in there?
So we did that not in an infection yet. We're also looking at that. But the way we did it was to use cytochrome transgenic cells and altered ligands. And so if you use a full agonist, they wind up pSTAT5 positive, and you basically saturate the Tregs. And so once you hit saturation, they can't control anymore, and those cells respond. But if you use a partial agonist, then it looks like the self-reactive cells. They don't become pSTAT5 positive, and that's also why they can't expand to a reasonable
And what
number.
if you use, you
At
can't.
102S.
And what if, do you have different TCRs that have different affinities? So can you look at,
We didn't do that experiment with TCRs with different affinities because we don't have a series with the same ligand. JL's actually going to build that for another reason in tumor models, and we could go back and check that. But Ermigarina published years ago that when you eliminate Tregs, then a population of low affinity CD8s emerge in the response that you didn't see if the Tregs were there. So it would only be repeating published data.
and one other question, when you were looking at the, in Ed's work, when you're looking at the localization of the T cells versus the B cells, within that B cell peak, there's a small peak of CD8s. Do you know, are those the CXCR5 or Ly108 CD8s within a
I'd
B cell
have to
follicle?
ask Ed for that particular point, but I would also, one of the things you have to keep in mind is something that we did a number of years ago. The B cells come out of HEVs, and the HEVs are at the periphery of the T zone, but they're in the T zone. They're not in the B cell follicles. So all the B cells, and this is something Hai had in his first paper about interactions directly out of the HEVs, the B cells have to run across a field of T cells to get into the follicle.
So,
And depending on exactly where you cut the lymph node and how you do that, even though we say CD4s are more peripheral, you can hit regions where you're enriched, for example, in CD8s, right at that border. The way to get around that, and we didn't do this experiment here, is to treat with anti-62L. So now you allow the B cells to clear the T zone. You get a beautifully crisp border at exactly where the FRC network essentially disappears when it's trying to go into the follicle, and that would give us a cleaner answer.
thanks so much.
Another one from home here. I don't know if you answered this, I'm sorry. Have you had any luck with CD-IBEX in bone and in tumor? Has this been attempted?
Yes. So in tumor, it depends on the tumor. We're working with the fact that the different tumors have a different level of necrosis and autofluorescence, and we're working to get around that. And we're also working with FFPE material since we think this is especially useful for humans. So preliminarily, we can get around that, but you have to do some things beyond what I've shown you to do that. And what was the other part of the question? Yes.
Thanks, Ron, for this great talk. Back to the Treg model. Of this handful of parameters that you said have to be very tightly regulated within twofold, have any of them been shown to actually, the change causes the autoimmune disease in patients, or
So
somehow
the
validated?
enhancers that Alex Marson has manipulated are loci of single nucleotide polymorphisms that matter
Okay.
for disease in human.
Okay, so they're all from GWAS and
Well, the mouse is not identical to the
Mhm.
human, but they're in relative positions where there are hits in the GWAS studies.
Uh-huh.
There's a very high density of GWAS hits in CD25,
Uh-huh.
so
Okay.
that's well known.
Okay, thanks.
We have 8 questions, but we're past the hour at this point, so let me just ask one more. Do you
Ask
think...
8 questions, huh?
Are
Fine.
you okay?
Yeah.
Everyone's okay? We're all set? Okay. Let's see what we can do. Do you think the shielding macrophages, quote unquote,
Right.
you talked about regarding tissue damage could be involved in immunosuppression at tumor sites?
Yes, they could be. These cells are, if you want to use a vernacular, M2-like, and therefore anti-inflammatory, and you want inflammatory responses to be anti-tumor. However, these, as I said, are embryologically derived cells, and most of the macrophages in a tumor have been shown to come primarily from monocytes that then enter and then polarize in this direction. So whether it's a similar process but not with these more resident cells, I can't tell you.
Okay, next. Have you been able to optically clear and use CE3D imaging in tumor tissue, and have you observed tertiary lymphoid structures?
In mouse tissues, that's ongoing, but it's been done. Again, we're trying to figure out the rules for why some tissues don't work so well versus others. It seems to be, we just had a big meeting yesterday about this, related to perhaps the level of necrotic tissue, which causes some unspecific staining, and the background level of autofluorescence. So those are some confounders. And most mouse tumors don't show tertiary lymphoid structures. They're predominant and dominant and visible in mostly human, and we're just starting to do human samples using this method.
Okay, not sure if this was addressed. In your tumor studies with anti-CD40 and subsequent conversion of Tregs to conventional cells, do you have any insights on whether the cells doing the conversion are naturally thymic-derived Tregs or more peripheral-derived?
So the best way I can answer that question is 60 to 70% of the what we call Tregs in the tumor volume are Helios positive. when we start before treatment, and that Helios goes away in these X Tregs, and actually from a substantial fraction of the residual so-called Tregs, which we think are in the process probably of converting. They just haven't finished losing all their FOXP3. So that would suggest that at least a large fraction of them would be thymically derived or conventional Tregs, but we can't rule out that there are P-Tregs in the populations as well.
Okay, almost done the lightning round here. We all benefit from you and your program's remarkable career. Thank you for the wonderful talk. In a given autoimmune disease-affected tissue, do you see a range of C-Treg-X Treg balance? And in organs, is there some spatial arrangement of these diverse states, e.g. along the gut or within the pancreas?
It's a very interesting question. We don't have a good method for addressing that in the human system, in human samples at the present, because we can't do the lineage tracing to identify an X-Treg. What we're trying to do is either mine existing data sets from single-cell RNA-seq or create such data sets that would allow us to identify a few marker proteins that we could address either by staining or by FISH or comparable transcriptomic methods to distinguish what was an X-Treg, assuming that this conversion doesn't change all the transcriptional profile of the cell. When we have that information, then we could do the types of experiments that are being asked to see what those balances and the distributions are in various tissues. We could theoretically do that experiment in mice with the lineage system, but we haven't done it.
Okay. Impressive images. 2 questions. I wonder if it is possible to capture cellular/tissue dynamics, including cell division / apoptosis, moving and interacting in 3D as well. And have you looked at T cell population in the context of solid tumors and their lymph nodes?
We're doing a lot of work on the tumors right now. Tumors are actually hard to image by 2P because by the time they're as large as you'd like them to begin perturbing them or doing treatment, they have a lot of limitations in light penetration and scattering that make 2-photon imaging difficult. There have been some good studies from Max Krummel and a few other labs, but many fewer than in normal tissues, so that's a problem. And to answer the first question, all of the 2P imaging is done in 3D, is done in a volume. The volume is limited by the speed of the cells more than the optical properties. If a T cell moves more than 1 cell diameter between each cycle of images you collect, then you can't trace the track or the path. And so that limits you to a depth of about 100 microns. But within that volume, yes, you can see interactions, you can see cells dividing, and we published on that. You can see cells undergoing apoptosis and fragmentation perfectly well.
Okay. What drives the sampling of the lumen in T cells in the gut? Are there microbiome-generated chemoattractants that are known to do this?
The answer is yes, in part. The studies that have been done on this kind of dendritic cell sampling suggest that there are signals to the epithelial cells that then make chemoattractants and may also change some of their adhesive properties to provide paths for the dendritic cells to extend their processes and do the sampling. But the details are not as well known as one would like, so it's a good question.
Oh, so Nina Shore. Very interesting talk. Tregs drain IL-2 to eliminate autoreactive T cells. Would you suggest anti-IL-2 therapy to treat autoimmune disease?
Unfortunately, no, because that also starves the Tregs, and if you starve the Tregs and kill them, then you've changed the balance again in principle in favor of the autoreactive cells. So if anything, what people are trying to do is to give a little bit of IL-2 in a form that favors the Tregs because of their higher CD25 expression while starving or preventing that IL-2 from reacting with the conventional cells so that you get Treg dominance over the conventional cells rather than trying to kill the conventional cells by starving them.
Okay, and then last one, it's from Ed Schramm, and it's the longest, so I waited in case you want to follow up with him. You mentioned that T cells follow a reticular network to reduce search times when encountering dendritic cells in the lymph node. But following these predefined networks might actually impede a cell's ability to follow the cytokine gradient, as when a regulatory T cell might prefer to make a, quote, beeline for the IL-2 producing conventional T cells, as in Hari's work. Are Tregs less tied to the reticular network, question, and or can T cells adaptively modulate when they adhere to versus disengage from the reticular network?
So we have not seen T cells move off of the network within the lymph node, except maybe when they're exiting through the lymphatics. But in terms of a beeline, what we have shown by direct imaging is that when a source produces a chemokine, in the case that we did careful work with a dendritic cell, but it could be the same cell that's making IL-2 if it's producing a chemokine by itself or through its interaction with a dendritic cell, which is what typically does this, that biases the walk on the FRC network. And although there are some vertices that you have to go through, the answer is it's a relatively very fast path, and you can get about an 8-fold improvement in finding the relevant cell compared to an equidistant irrelevant cell. So I'm not sure that it is a limitation of the system to do that. And Ed can follow up, obviously, in the lab, since we talk all the time.
Very good. Thank you.
All right.
All that's remaining, let's thank the speaker.